Cheers, thank you very much for the introduction and very much for the invite as well. So Fiona's done a tremendous job of telling us about the history of the GLP-1s and the receptors in general. And now I'm going to tell you about how we kind of figure out where the receptors are and how the drugs target these receptors in order to hopefully develop the next generation of therapies or improve current therapies. So I'm not going to go into this slide in much detail because I think Fiona's done a really good job of detailing the mechanisms of action. But suffice to say, we know really well how GLP-1 receptor agonists, GIP receptor agonists, triple agonists, etc. work in the pancreatic islets to induce insulin secretion. And this is really by conventional G-protein coupled receptor pathways. GLP-1 receptor is now a prototypical class B GPCR. It's G-alpha-S coupled, and once it's engaged, you increase cyclic AMP. That has a range of effects in the cell, and particularly in the beta cell you get sensitization of insulin granules for the release process, and that's a glucose dependent process. In the brain we also know quite a bit, to be fair, about where the GLP-1 receptor is located. We know less about how it signals in the brain, and of course where we kind of lack in information is in brain sites outside of the food intake, appetite regulation arena. So these are neurodegeneration, trauma and so forth. And then if we look at the heart, the kidneys, some of the other organs where we see positive effects of these drugs, we know basically nothing about where the receptors are located and how these organs might be targeted. So how do we go about looking at GLP-1 receptor and GIP receptor expression? Well, we use basic histopathology-based approaches. So we take a sample, we process it, and then we stain with antibodies that recognize the receptor, and then we can visualize where the receptor is. And it would be the same if you had a biopsy that went into an NHS histopathology lab. Now the problem is these approaches are not massively sensitive, and they're very non-specific, and particularly with GPCRs, these are low abundance proteins at the cell surface. They're actually quite hard to detergent stabilize, to raise antibodies against, and there's been a whole swathe of studies using antibodies that are not specific, which have really actually muddied the waters in terms of our understanding of GLP-1 and GIP receptor biology. This was an article by Lotte Hodson, who invented liraglutide, about the glucagon-like peptide 1 receptor or not, and about how we were maybe seeing it in sites where it actually isn't. And I think this comes on to sort of antibody companies. They're very happy to sell you antibodies and not actually do any of the validation, and when they don't work, they're not very happy to refund you. So it's really contingent upon you as a scientist to do the really rigorous work in terms of making sure what you see is actually what it is. So about 10 years ago now, we sort of were discussing with Dan Drucker and others about this issue. So we came up with a really simple approach of developing chemical labels for the GLP-1 receptor. And what this is it's an antagonist of the GLP-1 receptor, so extendin 939, a truncated form of extendin 4, which is Byetta. And we modified the molecule so that we could put various fluorophores or dyes on it, which would allow us to visualize the receptor both in vitro, in vivo, in live tissue, in fixed tissue, and so forth. And they worked really well. This is some examples. We made some knockout mice using CRISPR genome editing. You can see our ligands, which we term luxendins, will light up the islet in white. This in green is one of the only known specific antibodies from Novo Nordisk, but it doesn't work well in brain and some other tissues. And you'll see we don't see anything with both these approaches in the knockout mice. So at this point we knew these were good, and we've now distributed these reagents to hundreds of labs around the world that are using them to figure out where these receptors might be using FACS, imaging, pull down approaches and so forth. So most recently, of course, we have tirzepatide, which is showing game-changing levels of weight reduction. And tirzepatide is complicated because, of course, it has components of the GIP receptor agonism, GLP-1 receptor agonism, some glucagon components. And this really complicates our understanding of where it's acting, because it's now binding both receptors, is it? I don't think we fully know. Is it binding one or the other? Does it make the receptor dimerize? So forth, so forth. So it's a real challenge because actually tirzepatide, because of the GIP receptor component, shows massive increase in efficacy versus semaglutide, which is a single GLP-1 receptor agonist. So again, we used our proprietary technology to develop fluorescent tirzepatide or dual agonist labels, using a similar form where we put on fluorophores in the first instance. And you can see this works really nicely to now visualize GIP receptor, GLP-1 receptor, again, both in vitro and in vivo. And I should add, there's absolutely no known antibody for the GIP receptor, but if you go online, you'll be able to buy one. But those of us who know, we know. So I've taken you through a few of the labels, and we've done labels for lots of different ligands, and within reason we can do it for most of the current pipeline. But what I wanted to do is show you why it's important to know where the receptors are and kind of what we have learned from that. So if you give a patient liraglutide, semaglutide, tirzepatide, you will have resolution of metabolic liver disease without any worsening of fibrosis, or sometimes some resolution of that fibrosis, particularly with tirzepatide. So one would think, okay, these drugs must be acting in the liver to have this effect, to somehow defat the liver and counteract the fibrotic process. But we looked in all the compartments of the liver, we looked in the hepatocytes, all the parenchymal cells, all the stromal cells, all the immune cells in the liver, and we could not find the GLP-1 receptor. In fact, the only place we could find it is in gamma delta T cells, which are a vanishingly rare population of T cells, and that was only in unhealthy livers. And actually it was really nice, we got scooped by Dan Drucker on this, who also had very similar findings. So what this tells us is the GLP-1 receptor agonist, dual agonist and triple agonist going forward, or maybe not triple but the dual agonist, are working indirectly to have this effect on the liver. And it depends which colleagues you talk to. If you talk to an inflammatory colleague, probably has some immune cell component. If you talk to our metabolic colleagues, they'll say it's all about the metabolic normalization of the patient. Liver's a big sponge, right? So the more weight you lose, the more you defat the liver and resolve MASH. And MASH is a massive problem going forward, it's like a really big problem. So kind of shifting tack a little bit now, we do a lot of work in type 1 diabetes field. Type 1 diabetes, you don't have any functional beta cells left, they've been destroyed by the immune system, so you are insulin dependent. So recently Vertex have been running phase 1 trials of stem cell derived beta cells for transplantation into patients. And it started initially with around about 10 patients, 8 were still insulin independent following transplantation 1 year after transplantation. But when those transplants were salvaged in experimental medicine studies, the massive cells had decreased from 100% down to 8%. So there are issues with robustness and functionality of these transplants, and they also didn't lead to insulin independence in all patients. So we were quite staggered to find that actually the beta cells produced in this way don't really have much GIP receptor or GLP-1 receptor. And the reason that rang alarm bells to me as kind of primarily an islet biologist is because GLP-1 receptor is very beta cell specific. It's a mature beta cell marker, so if your beta cells are healthy, you have it. If they're not healthy, you don't really have it. And it's really important for the function because all the time you're producing GLP-1 when you eat, and it's really important, as Fiona showed, for proper insulin release and regulation. So what we did is we set up a pipeline and pathway where we produce these stem cell derived beta cells in-house. We use our loop senders to sort the beta cells into GLP-1 receptor enriched or non-enriched fractions, and then we can then reaggregate these cells then for hopefully eventual transplantation. And using this approach, what we see is with the GLP-1 receptor expressing fraction, we always enrich for maturity of the cells, and we decrease immature cell markers such as glucagon. So now going on to the brain, there's lots of GLP-1 receptors dotted around, but it doesn't really matter if the agonist can't access them, because if you inject a peripheral agonist, this is a peptide, it's got to get across the blood-brain barrier, or it can't. So where does it go? So these were studies we did in collaboration with Ben and Fiona, who are both in the audience, and Stefan Trapp down in London, where we injected animals with our luxendins and then tracked where they went in the brain. And what we saw is that the circumventricular organs, so these are parts of the brain without intact blood-brain barrier, absolutely light up. So the arcuate nucleus is where lots of the appetite regulating centres are. That was really brightly labelled. The hindbrain, particularly the area posterior MTS, that's probably where some of the side effects such as nausea come around. And this is really intriguing. This is the choroid plexus, and the choroid plexus produces cerebrospinal fluid that regulates your intracranial pressure. Turns out if you go into space, you can't regulate your intracranial pressure properly. Based upon this work combined with some experimental medicine work by a colleague, Alexandra Sinclair at Birmingham, these are now being trialled for prolonged space flight to prevent visual disturbance because of decreases in intracranial pressure, and they're also being taken forward by the US Army in terms of blast injury resolution, and intriguingly only Byetta works well for this, the other ligands don't. So now we're on to the dual agonists. So one might postulate that the dual agonists work better because they access the brain better, and kind of stands to reason, but doesn't. It's still a peptide, it still has a blood-brain barrier. And it turns out that actually it labels all the same sites as a single agonist in the brain, so it doesn't penetrate the brain much better, but it labels both receptors in those sites. So the synergy is likely because of the different neuronal populations now synergizing for the food intake effect. So we can really take a big zoom in now. So we know in the GPCR world that GPCRs are small. We can now see single molecules, and we see that the GPCRs are actually organised or clustered at the membrane of the cells. And when those GPCRs are clustered, it means they are signalling better and it contributes towards that signalling. So what we have most recently done is develop new GLP-1 receptor agonists which have a component bolted on which targets this clustering. And what we see in all the cases is better performance across the board for insulin secretion. Actually, we've got some food intake effects now as well, and brain penetration, because it turns out that the clustering moiety has an active brain transport mechanisms. So I think this is a nice summary slide because it shows you that when you know what the cellular targets are, and you understand the underlying biology, you can really kind of tweak and refine these molecules to perhaps perform better. And the argument is, do we need to make them perform better? Possibly not, but given the range of actions that these drugs are now starting to have, tweaking them for specific indications. For example, if you have Alzheimer's disease, you probably don't want to lose a lot of weight, might be an advantage. So I think this is where we're going in the future, is kind of tailoring and personalising these drugs for specific indications. So just going to finish on that. A whole huge amount of people contribute to these studies, but particularly the people that are up in the labs on the Churchill site at the moment, and happy to take quick questions or questions afterwards. Thank you.
Thank you.